going to know about genetics, unit number 2. First, the topic Mendel's work on transmission of trait. The way in which the characters are transmitted from one generation to the another generation. And it was first demonstrated by the Gregor John Mendel, and he is also known as father of genetics. And Mendel did all of his significant genetic experiments on garden pea, which is the botanical name of Pisum sativum. It fits all the criteria that makes an organism suitable for use in genetic experiments. And why did he select Pisum sativum? Let us know. Easy to cultivate and doesn't require much care. It is completely easy to cultivate. It is an annual plant, which is it will grow yearly. It can both self and cross pollinated, and it contains number of detectable contrasting traits like its size and pod size, pod color, like different detectable contrasting traits we can observe. He worked with about 10,000 plants, and reason for his success: selection of pea plant as an experimental material, restricted each experiment to one or few contrasting characters, tracked each character separately. Like if he did an experiment on one thing like pea pod size, like pod color, he only observed just one character like pod shape. Like in another one he only observed the color of the pea plant, like that. He had mathematical background, so he kept accurate quantitative data. The numerical data enabled him to do a rigorous analysis of hereditary transmission characters. He verified his results by further experiments such as F3 and test cross. Next, gene interactions. Let us know what are meant by gene interactions. The modification of the phenotypic expression like height, hair color, eye color, skin color. These are the phenotypic expressions. These phenotypic expressions of a gene due to the influence of the other genes is known as gene interactions, and there are two types: intragenic and intergenic. In intragenic, it takes place between the alleles of the same gene at same locus, and in intergenic we can observe that the interaction takes place between the alleles of the different genes of different loci. So this is the difference between the intragenic and intergenic. In intragenic we can observe these interactions of genes only occur on the same gene of the same locus, but in intergenic we can observe that these interactions occur on the different genes of different loci. Next, intragenic interactions. In intragenic interaction we can observe incomplete dominance, codominance and lethal genes. First, incomplete dominance, the condition in which both alleles of the heterozygote interact, influencing the phenotypic expression. Incomplete phenotype of the parents is called as incomplete or partial dominance. Here we can observe that this is the parent generation, this is the red flower and this is the white flower. When these are crossed in F1 generation, in their first offspring, we have observed that pink color has found. Like the red color is not completely dominated, like neither white color. They both crossed, and we observed that we got a pink flower. Then next, when it is test crossed in F2 generation, we can observe one red flower, two pink flower and one white flower. This is incomplete dominance. Codominance, the condition in which both alleles of the heterozygote show individual expression in the phenotype is called codominance. Like this, in this type of dominance, they have the equal share of their gene. We can observe that human blood group, they have IA and IA, and in B blood group it has IB and IB. Like IA and B, when we are crossed, we can get both IA and IB. They fused and got the AB blood group type. When A blood group and B blood group person get crossed, we can also get AB blood group. In this we can observe that the genes of both A and B are both sharing their genes, not dominating. They are sharing their genes, half of this, half of that. In F2 generation we we can observe that here A blood group 1 and AB blood group 2 25%, 25%, almost the 50% of the next offspring will be AB blood group and remaining 25% will be the B blood group. Next, lethal genes. It is also referred to as lethal genes or alleles that cause death of the organism that carries them. They are usually a result of mutations in genes that are essential to growth or development. The lethal alleles may be recessive, dominant or conditional depending on the gene or genes involved. We can observe lethal, which means the name denotes toxic, which means it carries, if any organism have this type of genes, it causes death to the organism. Most of these lethal genes can cause death to an organism, mostly in their early stages of their development. In lethal genes in mice, we can observe in parent generation yellow and yellow. If these are we can observe here capital Y and small y. Here capital Y and small y both are in heterozygote condition. We have gametes capital Y, each yellow mice has capital Y and small y. When they got fertilized in F1 generation, 25% of mice with homozygote condition, here we can observe homozygote condition in yellow color, capital Y. These type of capital Y of genes or alleles carries these type of lethal genes. That way 25% of the mice are dead, and in heterozygote condition yellow will be fine. And this is the non-yellow, which consists of small y. 25% of non-yellow will be survived, 0.5% of heterozygote condition yellow will be survived, but only the homozygote condition of yellow color will be dead in their early development of the stage. In their embryonic stage only they will be dead. Conclusion, we can observe that capital Y mice die. So in remaining 3, 2 of them are in heterozygote condition, that is which is here in the view of this one. He explained that yellow is the dominant character. So remaining 3, 2 are the yellow in heterozygote condition and remaining 1 is a non-yellow condition. Next, polygenes. What are polygenes? The polygenic inheritance is defined as a quantitative inheritance where multiple independent genes have an additive or similar effect on a single quantitative trait. Let us know about the clearly. If we take an example of height or skin pigmentation in humans, we find many different forms of 2 traits. We can't categorize like people in which 2 categories like tall and short for height. We can sometimes only in humans or anything, only we can't categorize only tall and short. They have the medium heights only. Like in height we cannot categorize into tall and short, and in color, skin color, we cannot categorize into dark and light. They have different types. We find continuous variation of these traits because their traits are controlled by multiple genes. This type of characters, traits like skin color, height, these are controlled by the different types of multiple genes. These are controlled. These are as many as 400 genes that control the trait of height and are responsible for variation in height present in the population. Approximately 400 genes are controlled. What are controlled? Height will be controlled by the 400 genes. The characteristics. Polygenes refer to a gene that exerts a slight effect, which gives a slight effect to the phenotype along with the other genes. Next, effect of a single gene is too small and it is difficult to detect. Multiple genes produce an equal effect. Each allele has a cumulative or additive effect. Next, multiple gene. Multiple alleles are 3 or more alleles for a particular gene. What is multiple alleles? The condition of presence of multiple alleles. An example of ABO blood group. We can observe that in human beings, or not only like in height, we can observe tall, short, and height. But in these multiple alleles, we can observe three types of blood groups: A, B, C. Sorry, A, B, O. Human genome, I means isohemoglobin, exists in three allelic forms: IA, IB, and IO. And IA, IB produce type A and type B antigens respectively on the cell surface of RBCs, whereas IO is a recessive allele and doesn't produce antigens. So, for instance, an offspring that got both IA and IB alleles will have blood type AB, and that who received IO would have the blood type of O blood group. If this one carries A and O, this person carries B blood group, contains B and O. Here we can observe A also present, B also present, O also present. In their next first offspring, we can observe one child get A blood group, and other child get AB blood group, and other child can get B blood group, and other child can get O blood group. Next, sex determination. Sex chromosomes are those chromosomes which singly or in pair determine the sex of the individual in dioecious or unisexual organisms. They are called allosomes. A sex chromosome that determines the male sex is termed as androsome. That is, andra means male and zoma means body. Example, Y chromosome in human beings. Basis of sex determination. Establishment of male and female individuals or male or female organs of an individual is called sex determined. We can establish what is the embryo, means what is that male individual or female individual is called as sex determination, and it is of mainly three different types. Here we can observe 5 types: environmental, genic, chromosomal, hormonal, and haploid and diploid type. Some organisms can exhibit environmental sex determination. In some of the organisms exhibit genic, some will exhibit chromosomal, and some will exhibit in hormonal, and some will exhibit in haploid and diploid. We can observe if some organism exhibit only in environmental, remaining they don't exhibit in this type of determination. They only exhibit in only one type of sex determination only. Next, sex-linked inheritance. Sex-linked inheritance is a biological process which involves the transmission of traits or characters from the parents to the offspring. X and Y are the two sex chromosomes that carries alleles at their gene loci. These traits that are being transferred from one generation to the another generation are present on autosomes, that is sex organs, the X chromosome and Y chromosome. The sex-linked genes are located on the X chromosome, result in X linkage. Similarly, Y linkage refers to the gene which present in the Y chromosome. Since females are homogametic with XX chromosome and males have XY chromosome, the Y-linked traits are transmitted via males only. We all know that females can carry their gametes. Their chromosome is XX. They are homogametic. In males, we can observe they carry XY, two different types of chromosome. So only the males can carry. They have two types. They have Y chromosome. That's why these inheritance related to the Y chromosome is transferred only through by males. There are three types of inheritance: X, Y, X-linked inheritance, Y-linked inheritance, and XY linked inheritance. In X-linked inheritance, certain sex-linked genes are located only on X chromosome, and their alleles are absent from Y chromosome. The alleles, the genes which are present on X chromosome, not in Y chromosome, only on X chromosome, these are transferred to their offspring. This type of inheritance we are called as X chromosome, X-linked inheritance. These genes are called as X-linked genes. The genes which are transferred from the parents to the offspring, the genes are called as X-linked genes. The characters which are transferred from the parents to the offspring is called the X-linked characters, and the complete pattern, this mode is called as the X-linked inheritance. The X-linked inheritance examples are color blindness. We can observe that some people cannot see the colors like red, green. So the type of blindness is called as color blindness, or hemophilia and the rhythm disorder. We can observe that this is the hemophilia. We can observe here. First, let us know it is the symbols of these diagrams. Is the XY unaffected male, which means XY is a male and XX is a female. If the white person is called as unaffected male, and here XX is called as a carrier female, and here XY red color, he is an affected male. He carries the hemophilia. And here we can observe unaffected female, and here the red color of the woman is called a female hemophilia. She carries hemophilia. These are the patterns of how the X-linked is transferred from. We can observe that X is both present in the father and mother. They both have X chromosome. So either mothers can be a carrier to send their disorder to the offspring, or they will be affected. Like father can also transfer their genes. Next, X-linked inheritance. The Y-linked inheritance. The sex-linked genes located on Y chromosomes are called as Y-linked genes. In the earlier slide, we observed that X-linked inheritance, the genes which are present on the X chromosome. Likewise, X chromosome is present in both the male and female. Here we are going to know about Y-linked inheritance, which is only present in the males. The Y-linked genes are confined to males only. Hence, they are called as holoandric genes. These genes are transmitted directly from the father to the son. Their mode of inheritance is called as Y-linked inheritance. Likewise, in Z, the genes which are transferred from the father to the parent to the offspring is called as Y-linked genes. The characters are called as Y-linked characters, and the mode of inheritance is called as Y-linked inheritance. Examples: hypertrichosis or ichthyosis hysterix. And hypertrichosis, we know that the hair will grow in any part of our body. This type of condition is called as hypertrichosis. The hair will grow in face, hands, they will grow enormously. So this type of condition is called as hypertrichosis. Here we can observe that this father is affected by if the father is affected by hypertrichosis and she is the unaffected mother, if they are crossed, then the both sons of their offspring will be affected by the hypertrichosis, and the females which are X type, they are present these type of X-linked inheritance. These type of characters, this type of disorder is only carried by this Y chromosome. We can observe this Y chromosome can only be transferred to sons. Females cannot be transferred. So that's why the disorder of this affected father will be directly transferred to their offspring of sons. The females will be unaffected. And XY linked inheritance. Certain sex-linked genes are located on both X and Y. Here we can observe, firstly we have observed the genes only present on the X chromosome. They are transferred from X chromosome to the parents to the offspring, from the parents to the offspring. In Y-linked chromosome, and as the Y chromosome is present only in males, through the Y chromosome they are affected from the father to the son. They are directly affected. But in this XY linked inheritance, these both chromosomes, the X and Y both chromosomes contains this type of genes which can inherit to their offspring. These are XY linked genes and their mode of inheritance is called as XY linked inheritance. Examples, the disorders like xeroderma pigmentosum, nephritis, retinus pigmentosum. Like xeroderma pigmentosum is their skin will be very dry, scaly, like that. If he is the carrier father and she is the carrier mother, one son will be complete. The one son will be the unaffected child. The two offsprings, like one girl and one boy, they both will be a carrier and one will be affected